Okay, so I'm here today to talk about bulk RNA-seq. If you're here to hear something else, then you're in the wrong place or at the wrong time. And I'm going to be talking about I'm going to give a little bit of a sort of background of the theory of how we do the analysis and sort of what sort of questions one might ask with bulk RNA-seq. Is this too loud? I feel like it sounds a little bit loud to me. You guys good? Too loud? Quieter? Yeah, that's what I was thinking. See if I can fix it before. Oh, he beat me to it. Okay. Okay, that's better, right? So next week, I will not be here. Dr. Pishu Shi will be talking about, so I'm going to be, like I said, I'm going to be giving background and then talking about the sort of bioinformatics part of the analysis. And then next week, Dr. Shi will be talking about the statistical aspects of the analysis. So this is, to some extent, kind of the boring sort of plumbing parts of it, but you need to do it right in order to have a good data set that you can work with. And I swear I fixed that and made RNA all uppercase, but apparently I didn't. Okay, so and I touched on this a little bit last week when we talked about high throughput sequencing. Since we're talking about RNA-seq, I'm going to zoom in a little bit on RNA-seq. So there are a bunch of different applications for RNA-seq. I showed you the poster last week. I think all of you, at least who are here in person, were here last week. And I'm not sure about on Zoom. So last week I showed you the poster of the 60 different variations on RNA-seq. So here's the high level view. What you get from RNA-seq is you're sequencing the transcriptome or some part of it. And so there are different questions that you can ask with that data. Basically you're getting information on which genes are being expressed in the given sample or samples that you're looking at. The most common application of that is for differential gene expression, saying, okay, what's different between sample A and sample B in terms of the genes that are being expressed or the levels at which those genes are being expressed. You can also use transcriptome information for genome annotation. I think I touched on that a little bit last week, and I'm not going to go into details. You can use RNA-seq data for identifying SNPs, at least in the transcriptome, right? You're not going to identify SNPs in non-coding parts of the genome, but if you're interested in coding sequence SNPs, you can get that from RNA. And you can also detect gene fusions, which is pretty common application in both cancer biology research and also clinically. This can be used to decide potential treatments. So today I guess today really we're not focusing on any of this because we're just doing sort of the back end part. But we're aiming for differential expression analysis, which is what you're going to be focusing on next week. I guess some of the things, if we're going to look at SNPs, we take a totally different approach from what we're doing today, or mostly different approach. And for RNA-seq, you can kind of throw everything into, one way to classify RNA-seq is either you're doing bulk RNA-seq or you're doing single cell RNA-seq. Bulk RNA-seq, which is what we are going to be focusing on today, means you're taking a whole sample with a collection of usually millions to billions of cells, and you're grinding it up, extracting the RNA, and then sequencing that. That's really hot. And so you're getting this consortium of expression, right? Even if there are different cell types in there, even if the cells are, in somewhat different states, you're just getting sort of a sum or an average of the expression across all those cells. There is a technology, single-cell RNA-seq, or a collection of technologies that we'll be getting to in 2 weeks, I think. Yeah, I think 2 weeks from today, we'll start talking about single-cell RNA-seq, and actually, I'll give the first lecture of that, and then we'll have someone, Tyler Shapey, doing, I think, 2 more lectures after that on sort of the hands-on analysis. So single-cell RNA-seq, you actually do get instead of getting an average, you get you actually get like per cell expression levels. And there are pluses and minuses to each. One of the big pluses of bulk RNA-seq is that it's way, 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 way cheaper than single-cell RNA-seq. It's also much less noisy because you're averaging over millions of cells. You just average out a lot of the noise that you tend to get with single-cell RNA-seq, both biological noise and technical noise. I will talk more about that in 2 weeks, because today I'm talking about bulk RNA-seq. So for the bioinformatic analysis, and I should say, if you have questions, please feel free to stop me. I'm hoping at this point you know that. And most set a timer, so I remember to take a break in a little while. Okay. So in a nutshell, our goal for the bioinformatic analysis is we're going to be getting sequence data, and so that's our input. And our goal is to take that sequence data and generate a matrix of counts where, depending on if you're a biologist or a statistician, either the columns are samples and the rows are genes, or vice versa. So this is what that looks like in practice. Here's just like a little snippet of the sequence data that you're going to get out of an RNA-seq experiment. This is like, the raw data. And then, again, our goal is to generate a matrix. Here, it's just, one sample, but you have each row is different gene. And this is the number of, we just have a matrix here where the values, the entries in the table in the matrix are the number of times we saw a read from gene D in our data set, the number of times we saw a read from gene C in the data set. Questions about all that? Okay. So to try and give a sort of high level way to think about how we're analyzing the data, I like to start off with what I'd like to call a biblioarchaeological analogy. And so imagine that we're archaeologists in 10,000 years from now or something like that, and we're excavating, we're studying these great ancient cultures that were in the city of Durham and Chapel Hill, North Carolina, and we're trying to understand, the societies in those great cultures. And so we're excavating at the former sites of the Durham main public library, and I don't think it's the main library in Chapel Hill, but some other library in Chapel Hill. And so what we're getting is like fragments of books, and we're hoping to understand something about a little bit about like what people were thinking back in 2024 or whatever, based on the books that were present in the public library and potentially differences between those libraries. And just in case you're thinking that I'm totally crazy for coming up with this example, this is to some extent a real field of research. This is part of a manuscript from Elephantine, Egypt. I have no idea where that is. There are other places where this has been done, but this is like part of a manuscript, and you can see like there are like little pieces of this. It's been pieced back together, right, from the little pieces, right? And so we're going to have data that's sort of like this, but a little bit cleaner, because as you can see, like these fragments are, like here's like a fragment with parts of 3 different lines. That's going to be really difficult to analyze. So I'm making things easier and imagining that we're actually getting fragments that are like lines of books as if they were like run through a shredder. I guess, yeah, run through a shredder horizontally. And so each line here is a separate fragment from a book. And each line is, again, to make things a little simpler, is exactly 100 characters. But they're not all from the same book, right? And so, like I said, we want to understand like what people were reading back in 2024 in Durham and Chapel Hill, and what are the sort of differences between those two great ancient cultures. And so our first approach is going to be to take this data, and we're going to search a database of, basically all books that were published, they were in publication in around the year 2024, right? Because this is just like, maybe it's interesting to read if you're into Dadaism, but otherwise it's just like sort of like random bits of text. And so you can imagine how you do that, right? Even if you don't know anything about computer programming, if I gave you like a whole database and I gave you infinite amount of time, or maybe like a whole bunch of undergrads, and even if like you didn't have a computer or anything, you could tell your army of thousands of undergrads, okay, you give this fragment to one undergrad and you say, okay, I want you to look through the database, and when you get to an O, stop and look and see if the next letter is a Y. And if the next letter is a Y, then look and see if the next letter is an S. And if the next letter is an S, look and see if the next letter is a space. And if you get to the O and the next letter is not a Y, just keep going until you get to the next O, right? So that's like a really simple way that you can search. It might not be the most efficient way, and it actually isn't the most efficient way, but it's simple, right? That makes sense to everyone? Does anyone like not make sense to anyone about how you could do that sort of searching? Okay. And so like the undergrad, you're going to say like, if you get to, if you've matched basically every letter and you get to the end of the line, like make a note on where you found it and then go find another fragment. So if we do that, and like I said, this is not like practically, well, first of all, like you wouldn't do this with undergrads. Well, I mean, unless you really want to like make a bunch of undergrads miserable. But, you could, there's programs to do this, and there are programs to do this a lot more efficiently than the way I just told you, but it gives you an idea. And so once we've done that with each of the lines, what we end up with is a list of the, for each fragment, the book that it was found in. And I didn't mention this before, but like we can see, we can keep track of like actually like where in the book it was found. And so there are different ways we could do this. Here it's, we're tracking it just by the character number, right? So if the first character in the book was 1, then this O here, maybe I'll indicate it here so people on Zoom can see it. So this O here is character 1,837,882 in Anna Karenina. Now, I should have asked if anyone could figure out where any of these came from. Oh well, too late. So that makes sense. How we went from like just text to a book and a position. Question about that? Okay, and so anyone notice anything funny other than my choice of books? Yes. So 2 of them are funny, and this was like a fortuitous accident. So I got these texts from Project Gutenberg, which is a nonprofit that they like scan in books that are no longer in copyright and they make them publicly available on the internet. And so if you download a file from Project Gutenberg, it includes a copyright legalese at the beginning that it's at the beginning of every file, right? And so I just randomly pulled these strings from the files, and just by chance, I got something that's not book, right? Or not like text, right? Each of these fragments, this one and this one is from one of these books. I don't remember which one. But it's not really book, right? It's sort of like, it's legalese, right? We don't care about it if we care about the book. But this is a perfect example because you get the same thing with sequence data. You're going to get like some garbage that doesn't like match the database. And for our concerns, like if we saw a lot of this and we didn't know where it was from, we might be worried. In this case, I looked at it and I was like, huh, okay, well, I know where that's from. And I don't really care about it because it's not biological, it's not book data. So I'm just going to throw it out. And as long as you don't have too much of it, that's generally what you're going to do for real biological data too. And so we're just going to kick that out. And now we have only book data. Okay, so the next thing that we want to do is we want to sort of quantify this and say, okay, well, how many lines, how many of these like reads do we have from each of the books, right? Because that was one of our goals, to kind of get a feel for like how much of each book there was in the library. And so that's pretty easy because we could do it one way is just like go through and like keep a table of like Huckleberry Finn and say, okay, there's one. Then Alice's Adventure in Wonderland, there's one, you know. We can also just like visually to make it a little bit easier, we can just sort them by the book that they're in, and then we can look and see like, okay, well, there are 3 from Ulysses, there are 3 from Alice's Adventure of Wonderland, there's 5 from Huckleberry Finn, and too many for me to count from Tale of Two Cities and Anna Karenina. But like ultimately we can take that and we can put that in a table, right? And that's, I told you that was like basically our goal for the bioinformatics part. So done, right? We started off with our snippets from the books, and we ended up with a table of counts. And so if we're just looking at this and looking at one of the libraries, like say this is from Durham, there's some technical issues with analyzing this and making assumptions like, okay, well, there were 3 times as many copies of Anna Karenina as Ulysses. So there's some problems with that, which I can talk about, but I'm not going to go into right now, because really our main focus for differential gene expression, and I'll say that for today, our main focus is in comparison. And so if we want to compare, then if we have data from Durham and we have data from Chapel Hill, we can look and say like, okay, it looks like we have about 4 times as much anachronina in Durham as we do in Chapel Hill. Now, there are some statistical issues with that just because the numbers are small, and we don't have like replicate samples or anything. But, you know, as a sort of first pass, you can kind of make that guess that, you know, about 4 times as much anachronina in Durham as Chapel Hill, about the same amount of Ulysses, about 3 times as much Alice's Adventures in Wonderland in Durham and Chapel Hill, right? Again, today our goal isn't to like do that, the comparison analysis, we're just aiming to get to this point. Questions about this so far? Okay, I'll keep going. Okay, so like I said, this is sort of like a toy example to kind of give you a feeling for what we're going to be doing with real sequence data. And it's very similar. So let's say we have some real RNA-seq data. Let's get a little bit more practical. We can break things down a bunch of different ways, but the way I like to think about it is that we have sort of 3 major steps. We have a pre-processing and quality control step. We have a mapping step, and then we have a counting step. And so, so first of all, the input for each of these steps is going to be the output from the previous step. And then here in this table, I'm showing what the format is for the sort of the output of each step. So the input for all of these, for the pre-processing QC is also a FASTQ file. We're going to talk a little bit more about what that is in a little bit. But so the, you know, pre-processing our input and output are FASTQ files. Then the sort of pre-processed FASTQ is going to be input to the mapping step. The mapping step is going to output a file in a format that's called SAM or BAM. I'll show you that later. Actually, I'm going to show you all these files later. And then that SAM or BAM file, almost always a BAM file, is going to be input to the counting step. And then the output of that is going to be just a table of counts. And that's usually in a CSV or a TSV format. Okay. So like I said, I will show you each of these formats. And for starters, I will show you, we'll look at a FASTQ file and kind of understand what information it has, like how it's structured. So FASTQ, first of all, the name comes from FASTA file, which is a file format that you might be familiar with. It's if you download a genome sequence or even like a sequence of just a nucleic acid sequence of a gene, it's almost always in a FASTA format. So FASTQ is short for FASTA with quality. I will show you what that means in a minute. And I just want to I have to give a shout out to one of my favorite Wikipedia pages in the universe, which is the FASTQ Wikipedia page. It's super helpful in as a reference for kind of figuring out, kind of interpreting FASTQ files. You know, I spend a little time every night reading it just to, I'm kidding, I'm not that crazy. But it is useful, and I do visit it somewhat often. Okay, so I already showed you a little bit of a FASTQ file before, but now I want to show you, I'm hoping that some people are familiar with FASTA files, so maybe even if you've never seen a FASTQ file before, the comparison will be helpful. If you've never seen a FASTA file before, it's okay. So there are a lot of similarities between them. So both of them have at least one header, and both of them contain at least one sequence in it. So for a FASTA file, the header is right here at the top, and and then there's sequence after it, and you can have what's called a multi-FASTA file, which means that you would have another header further down here, and then more sequence. So here, this sequence, you can tell if you look at the header, the structure of the header is actually like pretty simple. The requirement is just that it has this greater than sign here. And then the rest of it is sort of convention, but it's not a fixed standard. So like if you read this at all, you can kind of get the feeling that this is part of the sequence for an E. coli reference genome sequence. I have highlighting. I don't need to point. So FASTQ files also headers at the top. And the header, so for FASTQ files, FASTA files are almost always like there's some sort of human intervention involved. And so the header is usually intended to be meaningful. FASTQ files are almost always generated by a machine. And so the header is, not that the header isn't designed to be meaningful, but it's sort of like a little bit less informative. And we'll actually get through the details of what all this stuff is later. So that's the header. And then, like I said, both of them are going to have sequence. So for both FASTA and FASTQ, the sequence comes right after the header. And so then that's where they diverge. FASTQ file has this, actually two additional things. One is it's got this plus sign here, and the format requires either it can just be a plus sign or it can be a plus sign and then a copy of the header. Almost always you just see the plus sign, but sometimes you will see the copy of the header. Then this last part in the FASTQ file is what's called the quality score. And if there's time later, we'll get into details about how to read it. But I can tell you just sort of like what it means, which is that for each base in the sequence, each base gets a quality score, and that quality score tells you, if you know how to interpret these letters, tells you how reliable that base call is. So A is saying something about how accurate the sequencer thinks, how confident the sequencer is that's actually a C there, right? And for Illumina data, the best you get is about a 1 in 10,000 error rate, right? So the highest quality data you can get off an Illumina sequencer, there's a 1 in 10,000 chance that C is actually not a C. In this case, it's actually an A is, I'm going to say an A is like about 1 in 2,000, 1 in 3,000. I don't have it memorized, but it's something that you look up if you care. So for every base, you have like that sort of estimate of how accurate the base call is. Makes sense? Questions about that? Okay, and so we're going to use that quality information to some extent later on. And then I'll just point out like sort of the major differences is that for a FASTA file, the header starts off with a greater than sign. For a FASTQ file, the header starts off with an at sign. And then, like I said, you always have the sort of third part in a quartet of a FASTQ file is this plus sign. And I will say, just as an aside, you don't need to remember this too much. You'll notice that in the FASTA file, it's about, I can't remember if this is 60 or 80 characters on a line, and then there's just a line, a new line, and it goes on the next line. Right? That's just there for formatting. It's not really required, but it's pretty common to have those line lengths. It is pretty common for a FASTQ file for the line to be as long as the read length, right? So this is like one sequencing read. If you remember from last week, we talked about sequencing reads. So this is one sequencing read, and you're seeing the whole sequencing read. You'll notice that the line is longer than the FASTA file. The FASTQ file actually allows for the sequence to be wrapped on another line, right? So we could take this and chop it here and put the rest there. You almost never see that, but it is allowed. And people make the mistake in thinking that, okay, I'm gonna write my own program to parse a FASTQ file, and there are several mistakes that they make, and they end up getting their program ends up getting incorrect results just on sort of oddball files, and that's one place that you get mistakes. Another place that I'll tell you, just in case you decide you want to write your own FASTQ parser, is that thinking that you can search through the file for @ and that'll tell you all the lines. So @ is actually a valid quality character, and so you will see @ signs in the quality scores, and those are obviously not beginning a header line. That's a rookie mistake. Okay, so this is just showing you one read. Usually when you get FASTQ files, it's hundreds of thousands to millions of reads in a file. So it looks like this, continuing on down, miles into the earth at this scale, right? So each of these, you can see the @ sign, you can see the header after that, you can see the sequence, the plus sign, and the quality score. And so each of these is a separate read. Okay? Questions about that? Richard, you'll be proud of me that I remembered to tell people to sign in. One of these days, I'll have it all down. Okay, so I want to dive in a little bit deeper into the FASTQ file. So I just told you what the required format of a FASTQ file is: @ sign, header, sequence, plus sign. You can have the header again if you want, not required, and then quality score. And you have to have one quality character for every sequence character. Everything else that you see here is optional, is not optional, is not part of the format, but the manufacturers of the different sequencers have their own sort of conventions that they use. So this is an Illumina header. And so what the Illumina header, the convention is actually kind of cool. There's a ton of information here. So this first bit here, this M00698, that is the serial number of the sequencer. I think if I remember the 36 here, and this is where the FASTQ Wikipedia page comes in handy, because it explains all this. So if I remember correctly, 36 here means that this data was generated on the 36th time that this sequencer was run. This long string here, the 0000, whatever, AF, I guess all zeros, AFBEL, that's the serial number of the flow cell. If I was smart, I would have brought the flow cells back this week, and you could look on the flow cell, and you can actually see, at least for the next, NextSeq flow cells, you can actually see the serial number. It's actually etched on there, and there's a barcode on there that has a barcode version of the serial number, and you can actually look at the serial number, and if you look at the data that was generated by that flow cell, it's got that serial number right there. I think that's super cool. I cannot at all remember what that one is for. And then here, this next set of 3 numbers tells you where on the flow cell that this particular read came from. Now, remember that there's millions of spots on a flow cell. And so this and this are the XY coordinates on the flow cell. And this is, remember I told you that, sorry. Remember I told you that that when the flow cell is being imaged, it's captured, like it's a photograph, and then the confocal microscope scans over and takes another picture, and it takes another picture, and it scans and takes another picture. So so Illumina calls each of those like zones a tile, and it has a number assigned to the tiles. It's not that there are 1,101 tiles on this flow cell, but that's just how they have them numbered. I think this is actually the first tile on the flow cell. So this is like the tile or quadrant, and then within that image, this read came from, I think this is X and this is Y, but I could have it backwards. So like the X coordinate was 16483, and that's like the pixel position basically. And then the Y coordinate is 1412. Make sense? And then this other stuff is, I think the one here says if this is paired end data, then this is the first read or the forward read. And then the N is super confusing because this will either be an N or a Y. And I think if it's a Y, it means that, so there's some sort of like basic level of quality filtering that the machine does. And if it's a Y, it means it's for yes, it didn't pass the quality filtering. I don't know why they made that decision to make this N mean good and Y mean bad, but that's what they did. I don't remember what that 0 is, and that 0 has to do with the barcode. And actually, there's like subtle differences in the format. So like sometimes you'll actually see the barcode sequence here instead of a number. This data is a little bit older. So what you can do with this is you can kind of a little bit make sense of the structure of the FASTQ file. This is just a little bit of a snippet of the FASTQ file as it comes off the machine. If you process it, you can screw things up, but this is how it is as it comes off the machine. And what do I want to show you? Okay, so at the very top here, this is 2 reads from the R1 or forward read file. So this is paired in data. Actually, this is paired in data with a barcode, index barcode. So the R1 file is the forward reads. The R2 file is the reverse reads, and then the I1 file is the index, barcode index, and you can have an I1, you can have an I2 if you've got dual indices, or if you just have a single barcode index, then you just have I1 file. And so what can be a little bit confusing is that the first read in the R1 file is paired with the first read in the R2 file, and those two are then matched with this the first read in the I1 file, right? So this barcode goes with that read and that read, right? So this is the forward read, this is the reverse read, both from the same spot. And just in case you don't believe me, you can see that that's true because if you look at the coordinates for these reads, they're all the same because they all came from the same spot on the flow cell. And then if you look at for the second read in each of the files, these are slightly different, like the tile's the same, and I think this is a Y coordinate's the same, but the X coordinate's different. And this one comes from the same spot as this one, and from the same spot as this one. Does that make sense? So as the sequence comes off the machine, it's always, the file's in the same order. Now, we're going to talk in a little bit about how you can screw that up. And so you can't always assume if you have data that's been processed that the order will be right. But the coordinates, at least for an Illumina file, should actually be able to, you should be able to figure out like which reads go with which. Say that again?
And the y coordinate be same at x distance?
Yeah, because it's, you know, you so here's a flow cell. Those are the spots. We'll call this X, and that's Y. Right, so these two spots, maybe they have the same X coordinate, but different Y coordinates. These two spots have the same, wait, that's a, excuse my writing. I was looking, I was like, they're both X. I swear that's a Y. So these two spots have the same Y coordinate, different X coordinate, right? So yeah, you can, but they only, the reads from one spot should have identical tile X and Y coordinates. Right, so these are like unique addresses. Other questions? Okay. Let me see. Yeah, okay, let's keep on going for a couple more minutes. Okay, so that's a FASTQ file. So now let's talk a little bit about pre-processing and quality control. So I'm going to break down pre-processing into 4 steps. I won't talk so much about quality control yet. I'll just show you when we get to the hands-on part. So the first part of pre-processing is demultiplexing. So we talked a little bit last week about multiplexing. So demultiplexing, you are basically looking through, looking at the index barcodes and basically splitting each of these files up by barcode, right? So you end up with, I think I forgot to include my demultiplexing example here. But basically you're going to end up with, like you can see here, this index barcode is AGTTCC. This one is CCTGTC. You have a table that says this is from sample 3 and this is from sample 12. And so this read, this pair of reads is going to go into the sample 3 file. You're going to basically make new files per sample files usually. And so this read is going to go into the R1 sample 3 file. This read will go into the R2 sample 3 file. And usually once you demultiplex, you don't care about the index barcodes anymore, so you just throw them out. And then this read will go into the R1 sample 12 file. This read will go into the R1, This read will go into the R2 sample 12 file, and again, you can throw out the index barcode. These days, it's pretty common that demultiplexing actually gets done on the sequencer, or sometimes like if you aren't actually doing the sequencing yourself, if you're sending your samples to a company or to like a sequencing core, usually they'll give you back the data already demultiplexed. So we're not going to do that today because it's actually pretty rare that for RNA-seq data that I end up demultiplexing. So next step is quality control, which, like I said, I'm not going to walk you through it right now, but we'll the hands-on part we'll see what that looks like. And then after that is trimming and filtering. And I want to explain to you kind of briefly how that works. And I'll show you a practical example. And then maybe I think we'll take a break. So one of the things that you tend to care about in terms of trimming, there are really usually two types of trimming you're doing. One is quality trimming, and one is adapter trimming. The reason why you need to do adapter trimming is this. So imagine you have, I told you last week that you can have different read lengths, that's the number of cycles that you sequence. And depending on whether you're doing shotgun or amplicon sequencing, if you're doing shotgun sequencing, the fragment sizes are going to vary a lot, from sometimes even less than 200 base pairs up to somewhere around 1,000 base pairs. And so it is, if you're doing amplicon sequencing, then usually it's a narrower size range, although not always. But let's say we have a fragment here, whether it's amplicon or shotgun, where the insert, the sort of biological sequence that we care about is 230 base pairs, right? And that's just like a random number. So if we do 50 base pair sequencing, then we're going to sequence about this much of it. 100 base pair sequencing, we're going to 100 base pair reads, we're going to get about this much sequenced. 150, it's going to be about this much sequence. 250, it's going to be here. Now, 250 is bigger than 230. We're starting sequencing here, and so it means that we're going to sequence 20 base pairs of the adapter. The adapter is totally non-biological sequence. It's just there for technical reasons. We talked about it last week. And so if we just leave that there, it depends on how big of a problem that is, really depends on what we're doing downstream with the data and how good the software is at handling that. But in almost every case, if we know what the adapter sequence is, which shouldn't be that hard to figure out because we can usually just ask whoever did the sequencing. If we know what the adapter sequence is, we're almost always better off just looking for that and trimming it, bioinformatically trimming it. So we're not worried about like downstream steps, whether it's actually handling the adapter correctly or not. And like I said, sometimes the adapter, what's called adapter contamination, just having adapter sequence stuck in with the biological sequence, sometimes it can create problems and sometimes it really doesn't matter at all. So does this make sense? Questions about this, why we want to trim adapter? Okay. And so almost always the adapter trimming is going to be a sequence-based approach. We're going to actually look for the adapter sequence in our data. And so for trimming, here's kind of like an overview. There are a bunch of different decisions that you can make about trimming. This is just sort of like a normal set of decisions one would make. There's a couple different software packages for doing trimming and filtering, and they tend to like give you a lot more options than just what I'm going to show you here, but like this is sort of a reasonable set of trimming approaches to take. So we're going to be trimming based on 3 characteristics. One is we're going to be looking for quality scores less than 20. I don't think I mentioned this before, but those quality scores, they have a name. They're called Fred scores. And so a quality score of 20 is estimated error rate of 1 in 100. So basically, which is like actually pretty terrible error rate. So we're going to basically be trimming any positions that have a quality score of 20 or less, or I guess I'm saying less than 20. So less than 20, 19 or less. We're going to be trimming out N, and we're going to be doing adapter trimming. So here's our sample data that we're going to be trimming. So let's start off getting a little bit more tea. So let's start off and do the Fred score of 20. You will have to take my word for it that this 2 here, if you can see it, this 2 here represents a Fred score of, I think, maybe 13 or 14, definitely less than 20, whatever. I don't remember exactly. Again, something you can look up. And so we want to trim there. Now, when you're doing trimming, you almost always want to trim at the position like of interest and anything 3' of it. Now, the reason for that is that if we just saw this base and see, okay, well, this has a terrible Fred score, we're just going to toss that out. So we'll get rid of that A and that Fred score too, and we'll keep everything else. So if you do that, then you're essentially bioinformatically introducing 1 base pair deletion. And again, depending on like what sort of analysis you're doing, that could range from anywhere to, you know, mildly innocuous to like kind of disastrous. So generally, when you trim, you'll take that position and you'll trim everything 3' of it. So in this case, we're going to take that and everything, you know, everything 3'. And of course, this is the Fred score. We will not also trim the sequence. So after we're done with this, the quality-based trimming, it's going to look like this. Yep.
So you notice that then it starts off in like A, B, and C, and it goes down to lower letters or further letters in the alphabet, and you end up with like, you know, lower Fred scores, I guess. It seems like it's more chunk, it's really done I'm trying to justify getting rid of all that information.
And yeah, sorry, and that's a good point, which is another reason why you tend to trim 3'.
What different islands are really good or ahead of that? Can't count it 2 different leagues.
You're saying, could you like split it up?
Yeah, I think that would help.
You could. Yeah, you could. So how much you need to like kind of save data really, again, depends on, number one, how much data you have and what questions you're asking. So there are other approaches you could take. So one is that you could split the read and just take out the stuff in the middle. Whether that's useful or not, again, depends on the application, because like maybe having 2 short reads is no longer useful. You could also plug in ends for the low quality stuff. Whether that's useful or not, again, depends on the applications. There's some software that like just can't handle ends. And so that's our next step, is to deal with ends. So there are other ways to do it. This is the way that almost always you see. You can also do 5' trimming instead of 3' trimming, or both. So in theory, you could, you know, if it was low quality stuff at the 5' end, you could trim there. Almost always, like, you have tons of data and like throwing out some reads is not a big deal. And I will say that, again, a threshold of a Fred score of 20 is actually like pretty lax, because that's a 1 in 100 error rate, which for sequence data, keep in mind that if I had a 4-sided die that had A, G, C, and T on it, and I did my sequencing by rolling the die, my error rate is like 3 and 4, which is like, you know, not great, but that means I'm getting the base rate 25% of the time. You know, here you're a Fred score of 20, you're only getting it, you're getting it wrong 1 in 100 bases, which, you know, I don't know. I guess my die isn't so good, but.
So why does it tend to start off?
Okay, yeah. So that's just a function of the, so that's just a function of the Illumina sequencer. I've never seen like, it could be that there's an explanation out there, but I can give you my hand wavy explanation that I think is probably pretty reasonable. If you actually look at, a plot of, this is position or read position, and this is quality. For Illumina sequencers, what you tend to see is it's like kind of like that. So this is like maybe going out to like, it depends on the machine and what year, but let's say this is 250 base pairs. So there's like this little bit at the beginning that's like the first, say, that's about 10 base pairs, where it's a little bit lower quality. And this is like here is going to be a Fred score about 40, which is 1 in 10,000 error rate. And so out here, it's going to be, let's say about a 33 Fred score. And that's for the forward read. And then for the reverse read, it tends to be a little bit worse. You know, it's kind of tends to drop off later, earlier. And so my guess for the reason is like here at the beginning, it's just kind of like things are getting started. Maybe, again, this is a little bit hand wavy, but like maybe like you had a few spots where like it didn't, you know, the bridge amplification didn't work well or whatever, but they survived for a few cycles. And then as it's tailing off, I don't know if you remember from that table that I showed you last week, but for the newer machines, it's, I think, 3 or 4 days of running for like the longest read, like 150 base pair paired end reads or something. So you can imagine like after like all, you know, sitting there, like going through a bunch of cycles for a few days, like the, you know, I'm actually not sure if the reagents get refrigerated, if they're being refrigerated when they're on the machine or not. You know, the DNA is sitting there and it's going through, it's getting imaged, like the imaging probably, you know, the laser might be damaging the DNA. Like there are all sorts of things that can go bad, like towards the end. Again, kind of hand wavy, and I haven't seen a good explanation for what exactly is happening. But you know, as you go further in the cycle, and like I said, it does, you do get like this sort of reset effect when you go on to the, if you do paired end reads. But the second, the reverse read is never as good quality as the forward read, or on average, right? Does that kind of answer the question? Any other questions? Okay. So our next step would be looking for ends, and again, we'd probably we'd be finding any ends, and we'd trim at that point. Usually do 3' trimming. Ends tend to be pretty rare these days, and we don't have any in our data set. So we're just going to keep going, and we're going to do adapter trimming. And so we're going to be looking for that sequence. And if we find that sequence or part of that sequence, we're going to have some threshold, right? Because we're not just going to look for A or AA, right? Usually we're going to have a threshold of like, say, 8 or 10 base pairs. So if we find that, we're going to trim at that point in 3' of it. And so here we have a match. That's what, like 15 base pairs. So that's like a good solid match. So we're going to trim there. And so we'll get rid of that. And then this is what things look like after our trimming. And then filtering is going to be based on a few other characteristics. You almost always do the trimming first and the filtering. So filtering you might do based on length. You can imagine that if a read is below a certain length, it's kind of useless. So like, for example, a 5 base pair read is almost always useless, right? Especially if you're thinking of like, in the context of say like a human genome or even a bacterial genome, right? Any sort of 5 base pair sequence you're expecting to see lots of times, even a small microbial genome. You can also do filtering based on the sort of average Phred score across the whole read, right? So like even if like you don't have any like particularly bad bases, then you might have, actually I don't know why I chose that, but you might have, even if you don't have any particularly bad bases, it could be the whole read has sort of low average quality. And you can also like throw out, you know, reads that, if you have reads that have a bunch of ends, then that might indicate that there was some problem with that, some overall problem with that read. So here, the only thing that we're gonna, well, so here we'll start off looking at the length, and what's gonna show up is this read that we trimmed a lot. I don't know how long that is, maybe like 25 base pairs, but it's below our 40 base pair threshold, so we're gonna throw it out. So none of the other trimming that we did, you know, we didn't have any ends, and we didn't have, especially because we did the sort of Phred-based trimming, the quality-based trimming. And so that's the only trimming we have, sorry, the only filtering we have to do. We have one other step that we need to deal with, which is what I'm going to call maintaining synchrony, which is, I mentioned before that the data as it comes off the sequencer, it's, you know, the files, if you have paired in data, the files are in the same order. So if we threw out that read and we kept this read, then if we had software that didn't check to be sure that the headers match, which some software does and some software doesn't. So if the software isn't checking, then it's going to assume that this read, which is the first read in the file, goes with this read, which is now the first read in the R1 file. They don't have anything to do with each other, right? They could be from different samples. They're, I guess they're close on the flow cell, but that's just random. And so almost always we want to do our filtering in a paired end data aware, using software that's aware of the fact that paired end data exists, and will simultaneously filter the forward and the reverse reads, so that if you throw out a read from one file, you throw out the paired read from the other file. It is almost always the case that sort of software that does trimming and filtering does this, but there are some older software packages that don't. So it's just something to be aware of. And so we're going to get rid of that. And then after our trimming and filtering, it's going to look like this. Oh, sorry. Yeah. So we don't have anything to filter based on Phred score. We don't have anything to filter based on ends. And so this is where we started. We're going to trim there and there. We're just kind of rehashing a little bit. Trim there and there and filter, and we end up with this. And that's trimming and filtering. And now it's 2:12, and I went way longer than I planned. So how about we take a 10-minute break? 2:22, I guess. Or I guess any questions before I take a break?
Is 40 usually the cutoff?
It really, so it depends on the application. 40 is still a pretty good length. I think that actually the software that, one of the software packages that I use, I think has a default of 35 or 32 or something like that. I mean, 40 is still, I can't do the math in my head, but like, if you want to estimate, it's going to be a little bit wrong, but if you want to estimate how often you expect to see a sequence in a genome, like it's 4 to the n, where n is the length. So like a 40 base pair sequence, you expect to see, every 4 to the 40th bases, which I'm pretty sure 4 to the 40th is way larger than any genome. I think that's probably in the trillions at least. So that's a sort of state. Now, again, that's assuming that genomes are random, which they're not. Yeah, so if you're in a human genome, sometimes even 50 base pair reads aren't enough to uniquely position them in the genome, but for most reads, it's 40 or 50 base pairs is pretty good.
To look at, no.
Okay, well,
I'm just trying to think about like proteins in the body that would be, encoded by, something that is that small. Like
well, and so, but keep in mind that the reads here the read length is totally determined by the machine, and it's not, right, it has no sort of biological meaning really. Right, it could be, especially for DNA, right, it could be that the read is intergenic, it could be, intronic, exonic, at the junction of an intron and exon, at the junction between exon and, 5' UTR, 3' UTR, right? It doesn't, especially for DNA-seq, you're just randomly shredding the genome. And so like there's no sort of biological significance to the length of the fragment. For RNA, it's a little bit different because, right, it's like if in fact you do have like a very small transcript, then like insulin is really small. I mean, I think insulin's 100 amino acids or something like that, right? So then I don't actually know in humans if there are any introns and insulin or it's pro-insulin, right? Because I think it gets processed. I don't know. Anyway, yeah, but there's a little bit of it is limited by the length of the messenger RNA and things like that. But still, even then, and you're going to get like, if it's mRNA, you're not going to get any introns and stuff, but you're still randomly fragmenting. Most mRNAs are bigger than the read length that we're using, even if 250 base pair reads. Okay. So trimming and filtering, done with that. Questions about trimming and filtering before we keep going? All right. So let's talk about mapping. And mapping is kind of straightforward. We did that before, right? And it was basically taking that sequence and searching a database. In our case, the database is going to be like, almost always it's going to be a reference genome, right? So like if we have human samples, we're going to use the human reference genome and search our reads against the human reference genome. Like I said, there's, this sort of like simple way of searching where you like go character by character. That works. It's not the most efficient way, but so like the software we're going to use actually uses a more sophisticated algorithm. And there are some details that I'm going to talk about in a second that we need to think about if we want to kind of more efficiently use our data. But basically, we're going to be taking our sequences and we're going to be searching in our database, and what we want to know is where in the genome do they come from. And you can think of here, like the books is basically a chromosome, and then the position here as a position in the chromosome. And then then we'll get to the next step in a minute. Everyone okay with that? Okay. So there are some details that, because we're talking about RNA-seq, there are some details that we have to worry about. One is the fact that, at least for eukaryotes, we have introns, right? So we can't just take our read or if we just took our read and tried to map it against the genome, we would end up with things where, like here, this is a read, and this read is partially coming from exon 1 and partially coming from exon 2. And in the genome, there's this intron stuck in the middle. In the read, because it's mRNA, that's been spliced out. And so if we just naively sort of search the genome, maybe this would be enough to map it to the right place in the genome, or this would be enough to map it to the right place in the genome. But if we account for the fact that we know that the genome has introns and we know where those are, we're going to do this more accurate, right? So we're more likely to find actually where that went from in the genome. And so one of the approaches that, and I should say that the software that we're going to be using is called STAR. So it's specifically designed, there's other software that we would be using for if we were just working with DNA. But STAR is specifically designed for working with RNA, right? And so there are a couple of ways that it's going to deal with this. One is that it's going to take not only the genome sequence, but annotation of the genome that tells it where introns are. And so one thing it can do is it can just take that annotation data and it can take the genome sequence and it can basically make an in silico transcriptome, right? So it can splice up the genome into just the exons and search against that. So that's one simple thing. There are other tricks that it can use. For example, if part of a read maps and then the rest of it doesn't, then doesn't map for the continuous part, it can try and like remap this second part. And so that's what happens if you have, if you have an intron that's not annotated, then it can handle that, and that can actually, like, especially if it's like a genome, for human genomes, this isn't really much of an issue, but like if you have a genome that's like where the annotation is like only so, there might be some introns that you don't know about, and so it can actually identify new introns. One of the other big issues that needs to deal with RNA-seq data is, of course, we have these in eukaryotes, we get the poly A tails where you get a homopolymer of A's stuck at the 3' end of messenger RNA, and those aren't encoded anywhere in the genome, right? So if you just take the read and you try and map it against the genome, and it happens to be a read that includes a poly A tail, you're gonna have this string of A's that's not gonna match, right? So if you design the software such that it recognizes that there might be sequences, part of the sequence might not match the genome, and part of the sequence might just not match anywhere in the genome, it's going to do a better job of dealing with that. And then the third issue that is not really specific to RNA-seq, it's always going to be an issue with whether it's DNA or RNA, is you do have mismatches, right? And before, like I said, our simple algorithm was that if we got to a point where we had a mismatch with our database, we just skip it and keep going. But we're going to have mismatches for two reasons. One is that we'll have sequencing error because sequencing isn't perfect. And two is that the genome that we're using is almost certainly not, the genome that we're using as a reference is almost certainly not the genome of the organism from which that sample came from, right? Because no two humans, except identical twins and even identical twins, no two humans have exactly the same genome. And the reference human genome is like a mixture of like 6 people. Nobody or almost nobody knows who those 6 people are, but almost certainly any human samples you're working with didn't come from any of those 6 people. So there can be mismatches in part just because there's genomic diversity. So you can get away without doing any of this, but you're going to lose some data. And so it's usually going to be a good idea to use software that can handle these different issues. And so that's what we're going to do. I'm just going to set a timer so I remember to take another break in a little while. And so, like I told you, so that's what mapping is, right? It's just finding where in the genome a given read came from. And the output of that is going to be this SAM or BAM file. So SAM is short for sequence alignment map, and then BAM is just a binary version of the SAM file. I'm going to show you what that looks like in a second. Here is, I don't even remember if this is the most recent version, but this is the specification, version 1 specification for SAM files. It's not as good reading as the Wikipedia FASTQ page, but sometimes I need to read it. So I think I said this, but I just want to say this again in a different way. So, so a BAM file is just a SAM file that's converted to binary, and there are a few advantages to that. One is that BAM files tend to be a lot smaller than SAM files, and 2 is that it's much faster for computers to process a BAM file. So there's some software that will generate SAM files directly, and then there's other software that can convert between SAM and BAM files. That conversion process is like, it's a perfect conversion process. You can go back and forth between SAM files and BAM files. That represents exactly the same data. It's just that the SAM file, a human can like actually look at it as a text file, and a BAM file is all, you can't look at it directly. You need software to interpret it. But so some software will generate SAM files and then you convert it to BAM files. Some software will generate BAM files directly. It's almost never useful to keep SAM files laying around just because they're huge and they're not really useful for direct analysis. So I'll use SAM and BAM kind of interchangeably. The information that's contained in a SAM file is, it's got all the information. Actually, I should say what's in the SAM file depends somewhat on the software that generated it. So often, a SAM file will contain all the information that was in the original FASTQ file plus some additional information. So it'll have the header from the FASTQ file. It will have the sequence itself. It'll have the quality scores, and then it'll have information like based on that sort of mapping process, right? So that where the read mapped to, like which chromosome, what position, whether they're insertions or deletions or mismatches between the read and the reference genome. If you have paired in data, it can actually figure out, or it can estimate what the insert size was of the original fragment. Did I cover everything? Oh, it does have also kind of like the Fred score. It does have an estimate of how accurate the mapping is. And that, how it's generated, it really depends a lot on the software. And so that plays into like how you interpret that. So just to give you an idea of like what you can represent with SAM or BAM file. Actually, I think I can go through this quickly because I want to have time for us to do some hands-on stuff. So here, this is a reference sequence, and these numbers are positions, right? So this is position 1, 2, 3, 4, 5, 0. So this is like 10, 11, 12. So reference sequence, and here just a selection of reads. So some of these reads, like this R00/1 and R001/1 and R001/2 are, this is a pair of reads. Then there's some other funky stuff that we can, these are like sort of different weird reads, and we can represent those with a BAM file. So like I said, that's a read pair. This is a chimeric read. So you can see like this part that's in caps here is mapping there. And then this part that's in caps here, which represents this part that's in lowercase here, this is mapping there, right? And so it looks like either like in preparing the library or somewhere in the genome, there's some structural rearrangement. Here, this is a split alignment. So this is like, you can get this different ways. One way is if you have RNA-seq data and you're just mapping it directly to the genome, right? So that could be an exon right there. I'm sorry, an intron right there. And so all of that we can represent. This is a sort of a little bit of a toy version of what a BAM file looked like. So this is here, the first column here is going to be the name from the header. This is some information, some like, it can tell you different information, like whether it's a forward read or reverse read, or whether it's like split maps, other things. This is the name of the chromosome. So here, our chromosome was just called ref. So that's what that is. This, I think, is the position of the first character. Let's check that. So R1, the first character is 7. Yeah. So this is the position of the first character in the read. This is that. Do I have that right? I think this is the mapping quality, right? So that's how competent the software is. This is what's called a cigar string, and actually you can use this to take the read and kind of reconstruct. I can never remember whether it's read-wise or reference-wise, but basically it gives you instructions for going from one to generate the other. So this one here is pretty simple. The 9M means you had 9 base pairs that match, right? And so it's just a simple read. You know, the 9 base pairs in a row, they're a perfect match to the genome. When you have other things like an S, that's the 3S means you skipped 3 bases, and then you mapped 1 base. And I actually have no idea what P is. I is an insert and D is a deletion. And then this other one here is basically you're trimming off. It's skipping, but it's a little slightly different. And there's some other information here. Here's the sequence itself of the read. And it's not showing you here the quality scores, but normally in a BAM file you'd have that. And then this is the estimated insert size. Remember I told you it can kind of estimate based on where it maps in the genome. Questions about this? You don't need to memorize it. I just want to give you a feel for what information's in the SAM or BAM file. I'll be testing you on other stuff, but not this. Okay. So counting is also like kind of straightforward. We saw this with the library example, right? We just like take the reads and we look and see like how many, in this case, it's to like each, book, but you can imagine that instead of going by book or chromosome, you could go by gene. And so for that, to go to genes, you actually need the annotation of saying like, okay, this position here in this chromosome is in gene ABCD, right? And so what you end up with is a table where instead of books here, you have genes, right? And so we saw three reads from gene A, 9 reads from gene B. Of course, it's never quite so simple. Here is showing a couple of like pathological examples. So here's what you're normally hoping for, right? This is the gene, and you have a read falling totally within the gene, but you kind of weird stuff like a read, well, it isn't super weird, but the read is like partially in the gene, partially not. Here is a read that spans an intron, which, you know, for RNA-seq maybe doesn't make sense. Here's a read that's like spliced just like it should be. Here are two genes that are partially overlapping. So like for this one, that read, like you can say, okay, well, that's gene A. But for this one, is that gene A or gene B? And for this one, it's falling totally within like the gene A, the overlap between gene A and gene B. And again, you have to like decide, okay, well, is that gene A or gene B? There are different sort of ways that you can, different sort of rule sets that you can use. It turns out that you don't actually, most reads aren't so like sort of pathological, so it doesn't actually matter that much which one you choose. But there are, I have seen cases where weird stuff like this can actually create problems. Like for example, a lot of software, if it's like this, will just throw those reads out, but sometimes those actually are interesting position, you know, interesting data that you miss out if you don't realize that that some of the data is getting thrown out. Okay, and so I told you that the count table is usually there in like a CSV or a TSV format. So you probably all seen CSVs before, right? It just means that you have different, it's like a representation of a table where commas separate the different columns. Here is that same data as the TSV where you have a tab separating the columns. Questions about that? Okay, so I'm going to show you something really quick, but I think maybe we won't go into details of this now, and I can always come back to it later on if we have time after we do some hands-on things. So this is just a snippet of output from STAR. Actually, I should have mentioned before, STAR not only does the mapping, but it will also do this like counting step, which sometimes you don't even then care, you don't necessarily care about the BAM file if you get the counts out of it. And so this is output from STAR, and this is actually from one sample. And so these are genes, there's 4 genes. This is telling you the number of reads that were ambiguous. Remember, I showed you the ambiguous reads before. These are, no feature means that it maps somewhere that's not within a gene. And then multi-mapping means that a read maps equally well to at least 2 different places in the genome. The reason why there are 3 different count columns here and they look different is because there are sort of 3 main ways that you can make RNA-seq libraries. You can make an RNA-seq library so that you know which strand a read came from. And they're actually there are like about 10 different ways to do, 20 different ways to know which strand a read came from, whether it came from the read matches the mRNA or whether it's the antisense of the RNA. But all those different methods kind of produce data that fall into 2 different groups. One group is that the sequence you see like I said, it matches the mRNA sequence. So if the mRNA sequence, as you predict from the genome, is, GCGATGC, then that's the reads will actually have that sequence. And then the other group of methods generate antisense of the reverse complement of what you see in the genome, right? So if you see a sequence, it's going to map on the opposite strand as what you expect the minus strand for that gene. So either of these methods, it requires a little bit of one or two additional steps in the processing. Before these methods were developed, all RNA-seq data was unstranded, so you couldn't tell whether the read came from the sense strand or the antisense strand. So these days, it's pretty uncommon to see unstranded data. It's not like it's never present, but the additional steps to making stranded data are pretty minimal. And so like most library preps for making RNA-seq libraries generate stranded data. But you have to know whether the data is unstranded, sense stranded, or antisense stranded. So STAR will just spit out all three columns, and then in the sort of downstream processing, you need to know which column to use. We can do a little exercise later if there's time, or if we take some data and we figure out what it would look like. But like I said, I want to kind of skip ahead, or not skip ahead, but like move on to actually doing some hands-on stuff. So if you do not already have DCC on demand open, which now I'm realizing I should have done it. I should have told everyone to do it during the break, and I should have done it myself, but I didn't. So I'm going to open this. Oh, that's weird. Oh, I know.
Josh, before, there was a question in the Zoom chat.
Okay.
About, it was way back. I missed it. It's about for filtering and why you start with n, greater than 3 versus
So I don't have a great answer for that. I can tell you that one of the reasons why I have it that way in my slides is just because that is a default of one of the software packages that I use. And it really depends on the data and what downstream what downstream analysis you're doing, because some software just can't handle ends at all, and some software doesn't care. If you have like one N in a read, that's like, sometimes you get it, although these days, like I said, it's not super common. But I mean, this is a little bit of a hand wavy explanation. Right? So if you have, one N in a read, like it's not great. 2 N's is starting to look pretty bad, and once you get up to 3 N's, like that's a total mess. So I'd say that's sort of a like a one way of thinking about why you might want to say, I'm going to throw out any reads that have 3 N's or more. Okay. Anyone not have, I'm sharing that, right? Yep. Anyone not have DCC on demand open? Cool. Okay. So if you've been to any workshops before, you've experienced this, we're going to do things slightly differently. So we're going to go to the CHSI RStudio workshop. You can leave everything the same except for CPU, we're going to change it to 16. And for memory, we're going to change it to 64. And this is just going to let us do things a little bit faster. Actually, for the memory, no, actually, we could get away with less. We're going to use multiple CPUs at the same time. So the software star that we're going to use for part of this is able to use multiple CPUs in parallel. And so by using 16, well, we won't quite get a 16-fold speed up, but we will get some speed up. And it could, so star is, it used to be considered memory hungry because if you're using a human genome, you do need about 40 gigs of RAM, which used to be like an impressive amount of RAM, and now it's like, actually, before I talk anymore, I'm just going to get this started. So does anyone need to see this longer? So 16 CPUs, 64 gigs of RAM, or 64 for memory, and everything else you can leave it the same. Okay, I'm going to launch it. And this is probably more than, like I said, we could use fewer CPUs to just be a little slower. We could, with this data set, we could use less RAM. And hopefully it starts. And actually, while we're waiting for it, oh, there it goes. I'll tell you a little bit about this data set. I should have included a slide and I totally forgot. So this data is, it's fungal, it's from a fungal pathogen that is it's an opportunistic pathogen, which is why it's relevant for HIV/AIDS workshop, because it's actually one of the, so it's from a pathogen called Cryptococcus neoformans that basically only affects people who have some sort of immunosuppression. So it used to be, I guess still there are people have AIDS if they have HIV that's not treated or not controlled. So people with AIDS, people who have immunosuppression, either like inborn or due to, immunosuppression for a transplant or for managing some other illness or for, because they're being, treated for cancer. But if you have a sort of normal functioning immune system, it is extremely rare for people to have, to be infected with Cryptococcus neoformans. Wow, that was really loud. So that's the data set that we're going to be working with. And there are a couple of advantages of working with this data set as opposed to, say, like human data. One is that the genome is way smaller than the human genome, and so that makes things a little bit faster. And another is that we don't have to worry about patient privacy, because we don't care about that for our fungus. Okay. Does anyone need me to walk through how to clone the repo? This is the same repository that we've used for all the sessions since October.
You're just pulling it?
Well, so I'm going to pull it, but if you haven't cloned it yet, then I can walk through that.
Yeah.
You want me to walk through it? Okay. So I will go from my notes. Let's see. So if you've never, actually, let me just clean up. Ignore me for a second here. Oops. So actually, before I do anything, you're going to want to go here and actually probably follow along to some, oh, that was the wrong thing to do. So if you go here, either the QR code or this URL, that'll get you to the Git repo. And you are going to want to click on initial download of workshop content. If you already have cloned the repo, you should be able to just go in here. If you don't have it open as a project, click here and find it. And then, well, I'll show you in a second. Okay. Let's see, what am I doing? I need this. Okay. So you want to come here and you want to click on the code button. Oh yeah, I was gonna, hold on. Let me just do something really quick to clean up what I downloaded before. Okay. So you want to go here to this code button, click on it, and here where it says clone with HTTPS, you're going to click on the little clipboard next to it, and that's going to copy that URL. Anyone need me to do that again? Okay, so then you want to go back to RStudio. Does anyone need me to make this bigger? Yeah. And there are a couple different ways you can do it. I'll go here under the project menu and say new project. And you want to click on version control. Git. And then you're going to paste the URL that we copied. You're going to paste in here. So on a Mac, it's Command V. And on a PC, I think it should be Control V. You can just leave the create project as subdirectory of. If you have a tilde there, that's good. If not, you can click on browse, and probably the easiest place to do it is your home directory. You can click on home, and that'll just put a tilde there. Click on choose. And then for project directory name, if you click here and push like Control or Command, that'll copy the project directory name from the URL, which is usually the default. And then I click create project. Anyone need me to wait before I do that? Okay. Say that again? Oh, sorry, you want me to do that again?
We took it as workshop automatically. In the name of the subject.
Okay, hold on. Let me see. I mean, it's not the end of the world. So it's possible that it's for create project as a subdirectory of. Okay, so if you want it to do, if you want it to be in your home directory, I'm just doing it in home directory for, it really doesn't matter as long as you can find it, but if you want it to be in your home directory, which is what I'm doing just for simplicity's sake, you go to browse and then click on home here, and then should get you to your home directory, and it should have a tilde there, and then you can click on choose. I'm going to do cancel. Anyone else need me to do any of that again? Okay. If you've already cloned it and you just want to get the updates, you can click on, if you're in the project here, you can click on the Git tab and then just click on pull. So mine's saying already up to date because I just cloned it, but if you haven't pulled it recently, you should get some additional files. Okay, anyone not already with the repo? Okay, So we are going to go into assays. So down here in the file pane, go into assays, RNA-seq bioinformatics, notebooks, and this is where we're going to be, just to show you if you want to look in GitLab. So if you go to assay workshops, the bulk RNA-seq bioinformatics table of contents. So this is what we're going to be going through: the quality control, trimming and filtering, download and index genome, and mapping reads to reference genome. And then like all of this is kind of bonus material. And I'll just point out, we're going to be using, for actually, I guess all of the steps today, it's going to be mostly Unix commands. I'm not going to take the time, just because we don't have time today, to talk about Unix. If you want to kind of get a little bit of an introduction to Unix, I have here a link to this software carpentry, the Unix shell. So that's a lesson or a set of lessons. It's Introduction to Unix. If you want to, you should be able to run through that within RStudio on DCC on demand. And you do need, that assumes that you've downloaded some basic things. And if you click on this, actually, I should show you, I'll show you in RStudio. So this is a little R Markdown file that will download those files for you, but really you want to do it in here. And it is prep, I can't remember. No, I'm gonna have to find it. Oh, that's where it is. Okay, if you go to the top level of this repo, if you go in miscellaneous, Unix shell setup, and you really just only need to run this chunk here. It'll download that sort of those files and unpack it so you can go through the Unix demo. Does that make sense? I should actually probably include those instructions there. I'll try to remember to do that. Okay, so I'm going to go back. So assays, RNA-seq bioinformatics, notebooks, and the other thing that I'll mention is that all the data, the FASTQ files that we're using is already downloaded on DCC on demand in a shared directory. If for some reason you wanted to like run this on a different computer, there are also scripts in here for downloading the data, and like a few other sort of accessory scripts. We're going to be working through an example just with one FASTQ file. There are also different scripts for like running the whole thing on all the data. And I've done that so that those files are like available for next week when you're actually doing the downstream analysis. But today we're just going to walk through one file just to give you a taste of it. Okay. So the first thing that we're going to do is we're going to do quality control, which I kind of glossed over before. So for that, we're going to use this fastqc.rmd. And so this is we're going to be running two programs, one called fastqc, that's a program for kind of doing sort of quality control, generating quality control graphs and data for RNA-seq, or sorry, for any high-throughput sequencing data, any FASTQs. And then we're going to use another program called multi-QC, which if you have a bunch of FASTQ files, it generates sort of summary plots across sort of larger data sets. And so this first chunk here we're going to run is just setting up, it's assigning to a few variables the paths where we're going to find the input data and where we're going to put the output data. It's also checking to see how many CPUs are available, and it's going to use that information later. And I'm going to go through kind of quick just because we only have about 45 minutes left and we might even want to take a little bit of a break. But feel free to stop me. Like I said, I'm glossing over some stuff, but I want to give you like a little bit of a taste of what this analysis looks like. Okay, so the next step we're going to do here is we're just going to make a new directory for the output, for the quality control output data. And so that doesn't, when you run that chunk, it's, that's weird. Oh, I forgot to run the first chunk, which is the library, loading the FS library. Okay. So now when I run this chunk there, it should create output directory. And so this is a variable, this QCdir, that we defined up here. And then if we run file info, just to be sure that that directory actually exists, that's really all we care about, is that it's saying here that it is in fact a directory. And if it's a directory, we know that it exists. This next chunk that we're doing here is, so our Markdown is, and RStudio are originally primarily designed for working with R. But like I told you, we're actually going to be running most of this stuff at the command line. We're going to be using a shell that's called bash, which is, it's a shell, which is a kind of a program that lets you run other programs, but it's also a programming language. That's a whole other conversation. But we want to be able to have some information available in the shell. And normally R and bash don't communicate directly, but we can use this function syssetend, and it lets us define bash variables. So this chunk is an R chunk. You can tell that because it says R right there, but it's going to define these bash variables, raw FASTQs, QCdir, and num CPUs. And it's going to define them, it's going to assign values to them based on these R variables that we set up here. Okay? And so we're going to run that chunk. It doesn't, nothing obvious is going to come out of that, but it'll let us, in these chunks further down that are bash chunks, those variables will be defined. And so now we're getting into bash, and you can tell because here within the curly braces it says bash, so that's telling the knitting function that's running these R Markdown chunks. Okay, well, you should actually run this. This is bash code, and this is not R code, so you should run it as if it's bash code. So the first thing we're going to do is we're going to just call this program fastqc, and if you call it with an argument of -h, then it gives you help information. This is a sort of standard in the Unix world, is if you run a program and then you follow it with either -h or --help, it'll give you a little bit of information on how to run the program, sometimes a lot. So here here's some information. It's actually a lot of information. It tells you that if you want to run the program, you call fastqc, and then you give it sequence files. And then there are all these different options you can give it. Like you can tell it where to output the results. You can tell it, let's see what else is interesting, how many CPUs to use, the -- threads. There are a few other things that you can tell it. -- extract, which means that it's going to, by default, it'll output this as a zip file. It's a compressed file, but we're going to just tell it to not bother to compress it because we're going to want to look at those files. So here's the command we're going to use. I'm going to start that running because it'll take a minute. We're just going to ignore this set-u for now. We're saying run fastqc, run it with, you have this number of CPUs available, so you can like actually run it in parallel. And that, remember, we define that further up. Like I said, this extract thing is telling it, is telling it that we don't want it to bother to compress the output. This is the input file. It's this sr129336660.fastq.gz. That's the gz means it's G is short for gzip. It's another type of compression. So this is a compressed fastq file. And then this raw fastqs is a directory that we also defined further up. And then this is saying output it to this QC directory that's also another variable that we defined further up. I know that's a lot. Questions? Yeah.
I'm just kind of curious as to why, like, running this through R is preferable to doing this in like command line, or through like a JavaScript or something like that.
So the reason, if you were here for the very first workshop, which I don't remember if you were or not, I talked about reproducibility. So the reason why it's preferable to do it in R Markdown as opposed to just at the command line is for exactly that reason. Because we have this document here that we can not only run it, but we can come back to it and run it again. We can give it to someone else and say, this is how we ran it, right? Whether that's an R Markdown document or a shell script or like any number of other, there are a bunch of other ways that we could do this reproducibly. That's a matter of taste. Sometimes I like doing stuff in R Markdown, especially when I'm sort of like developing a pipeline. But sometimes, like if you look at the example, one of the versions of the scripts that I have for running all the data is actually a shell script it's a Slurm batch script that it basically runs on all the data in like 2 or 3 minutes because it will distribute the jobs across the cluster. So that decision is kind of, that's a matter of taste. I'm certainly not going to tell you that the only way to do this is in an R Markdown document. I like R Markdown. I think it has some nice benefits to it. But I will tell you that doing it within some sort of script is way better than doing it at the command line. Yeah. And doing it, better than like typing things at the command line, because then you just don't, you don't have any sort of reproducibility. Yeah, and also you can't then come back and figure out like, okay, what did I do last week? Right, whereas this, like you can do that, and you can also like next time you want to do this for a different data set, you can have this as a starting point. And yeah. Other questions? So the default for fastqc is that it tells you as it's going along, it's 5% complete, 10% complete, 20% complete, right? And then once it's done, it'll say analysis is complete. Okay, so we can take a quick look at the results. And this will tell us where to find that. This is just like printing the value of that directory. Okay, so I'm gonna find mine. If you just ran it, you should be able to find yours a similar way. So mine is in my work directory. Yours should be in work, your net ID, HIV 2023, RNA-seq QCDir. Okay, so let me show you how to find that. If you click on these 3 dots here, that will, oh, actually I did it wrong. The easy way to do this is going to be to highlight that and then copy it. So on a Mac, Command C, on a PC, I think Control C works. then click on the 3 dots and paste it. So on a Mac, Command V, on a PC, I think Control V works. Anyone not there? And of course, you can do /Josh, /work/Josh, maybe, but better to put your net ID in there.
Where's the 3 dots?
Huh?
Where's the 3 dots?
They're here in the file pane, like right at that next to the R cube. Get it? Anyone need me to wait? Okay. And then if you click OK, then basically the file pane is now going to zoom to that directory. And so what we're looking for is this srr blah, 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 fastqc.html. So if you click on that, RStudio will give you the option to view it in the web browser or open an editor. We definitely want to view it in web browser. And so this is the fastqc output. And it gives you some basic information like the input file name. It tells you the quality score encoding. I'm not going to get into that. How many sequences there were, the average sequence length, the percent GC. And then this is summary. So here's the summary on the left-hand side. And sort of the short way to think about this is if you see a green check mark, you're probably good. If you see an orange exclamation point, there might be a problem, but there might not. And if you see a red X, there might be a worse problem, but there might not. So this is the per base quality score. This is sort of like that figure that I drew, right? This is the x-axis here is the position in the read, and this is the average across all the reads. You'll notice that it sort of bins some things in the center, right? So this is right here is positions 36 and 37. So here's the quality score. You can see that little shoulder like I showed you before. It's very sort of slowly dropping off. And very end, there's a bigger drop off. And then it gives you error bars on those qualities. So you can see that the quality scores average at the beginning, they're starting at about 32. They kind of go up to 35. And these are 76 base pair reads. So the last couple of bases, it's dropping off a little bit. And remember that, so a Phred score of 30 is a 1 in 1,000 error rate. 40 is 1 in 10,000. So these are pretty good qualities. And it gives you, the green zone here means it's pretty good. The yellow zone is so-so, and the red zone is low quality. Here is the average. So this is positional average across all the reads. This is the average taking all the quality score for each read, taking all the quality scores, averaging it, and then basically a histogram, right? So most of the reads have an average quality score. Vast majority of the reads have an average quality score of about 35. So that's good. This is again, averaging across all the reads, but looking positionally. And this is the red is T, green is A, blue is C, and black is G. And it's showing you the percentage, the base content for each position. This part is exactly what you're looking for. So if the GC content of the genome was 50%, you'd expect in perfectly good data, you expect a flat line. You expect all the lines to be laying there on top of each other. It's not. We saw at the top that the GC content is 52, which is probably about right for this genome. I don't remember. Right, and so there's you expect to see a little bit more G and a little bit more C, right? And that's what you see. And then Otherwise, here it's flat. This part at the beginning where it's not flat, that indicates that there's some sequence bias. Ideally, so theoretically, there's no reason why you should see that, right? It should be that because you're randomly chopping up the genome and you're just sort of like randomly sampling. In reality, you almost always see this. I've never seen a good explanation for this, but I have hand-wavy explanations, which is that there's probably a little bit of sequence bias in terms of fragmentation. There's probably a little bit of sequence bias in terms of the reverse transcriptase. There's probably a little bit of sequence bias in terms of the ligase that's ligating on the adapter. So you probably do have like a little bit of sequence specificity that ends up at different stages in the library prep. You always see this. I'm not concerned. It's giving you like a little bit, it's giving you like a orange exclamation point, but I'm not super concerned. You also, here's the like distribution of GC content, just like a theoretical, well, so blue is theoretical and red is what you're actually observing. So it's a little peakier than the theoretical distribution. I don't know what to do about that. Like, it is what it is. Yeah, that'd be great. It never does exactly. Like, I'm not super concerned. I should say, actually, in general, for the QC, for the most part, for most of the information, we're looking at it and we're going to say, you can't really do anything with the information. The one thing there are two things you can do with it, which I'll tell you in a sec, or I'll tell you one of them in a second. The other one is you can look at it and you can say, this data is a pile of crap. I gave like high quality samples to company sequencing or whoever did the sequencing, and they give me crappy data. And, I'm going to ask them to run it again or give me a refund. Right? That's the big thing you can do with this data. And occasionally, any sort of most respectable organizations that provide sequencing will actually do some sort of QC on the data before they give it back to you, and will, if there's a problem, will rerun it themselves and just, let you know, oh, it didn't work so well the first time, so we ran it again. But, sometimes that doesn't happen. Again, here we're looking at the like across the reads average N content. Like it's basically, you can't see anything there. I'm sure there's some N's in there, but there's nothing really concerning. Here's the distribution of sequence lengths. Almost always when it comes off the machine, all the reads should be the same length. And this is, all of them are 76 bases. That's good. Sequence duplication is basically read sequences or like subsequences that occur multiple times. So for DNA-seq, this is a little bit of a concern because it should basically be a random sampling of the genome. For RNA-seq, we expect some genes to be expressed at higher levels than others. Especially for RNA-seq, you get a big chunk of the data ending up being ribosomal RNA. So you're going to get some sequence duplication. This is, FastQC says like, warning. I say whatever. You also get some overrepresented sequence here. And so for some things, FastQC will tell you what it is. Sometimes it can't identify it. So it's saying, oh, we have a bunch of TrueSeq adapter. That's adapter contamination. So this is the other thing you can do with it, which is in case you didn't happen to know, like here's the adapter sequence, we should trim it. And this is showing you just like the adapter content across at different positions. You can't tell that, that it should be, I think it's going to be the Illumina universal adapter. Yeah, I don't know. The universal adapter is pretty similar. Anyway, you can't really see it. There is like, it's somewhere, we're seeing this in 0.18% of the reads. or part of it in 0.18% of the reads. So like, you know, here, the scale here is 100%, right? So like, it's way down here. It's obviously not terrible. If you actually see the line coming off the x-axis, then, you know, it's worse. You can, so we're going to do, this is the raw data. We haven't done any trimming. We can run this again after we've done trimming. We're not going to just today in the interest of time, but you can run it again after you've done trimming. And certainly like the overrepresented sequences and the adapter content should be even, I mean, here, like we're not going to be able to see it, but it should be even better. Yep.
Why would it be at every position when you see this adapter?
No, so it's going to be, it just depends on how big the fragment is. So, or yeah, how big the insert is. So let's say we have let's say that's here. That's 500 base pairs. This is not to scale, but let's say this is 70 base pairs. This is 50 base pairs. And this is 30 base pairs. We've got our adapter. So for this one, this number here is the length of the insert, right? So for this one, let's say we do 100 base pair reads. For this one, the adapter is going to show up starting at position 30. Here it's going to start showing up at position 50. Here it's going to start showing up at position 70. And here you're not going to see any adapter, right? So it does give you a little bit of an idea of the, for this plot, it gives you a little bit of idea of the sort of insert size.
Is the insert ever going to be just 10?
It can be, yeah. I mean, it's, you know.
It is the whole like RNA.
This is the chunk of RNA, yeah, after fragmentation. It's chunk of RNA that got adapter stuck on it. Or for DNA, it's the chunk of DNA, right? So you get a distribution of sizes. I mean, it tends not to be that small, but you do get that. Okay. So let me switch back here, and I should have started this before. So I'm gonna start this chunk running really quick. So this is, I'm skipping a few just in the interest of time. This is gonna run FastQC on like 10 of the files, just so I can show you, and I have it running -- quiet, so it's not gonna output every step like this. Oops, what happened? Oh, it's already done. Okay, and so this is running on 10 of the files, just so I can show you multi-QC, which I will skip this step here, which, well, I'll run it. It's just like FastQC, if you run multi-QC with -H, it gives you some help information. So if you can imagine, here are data sets like 30 or 40 FastQs. You can imagine it would be like a little bit of a hassle to like look at this report for each of those. So multi-QC summarizes that. I'm just going to run it. And it just generates sort of one summary report, which is going to be here, I think. No. Oh, no, it's right here. Yeah, so it's in the same directory. You might need to refresh it, and we're going to look for this multi-QC report here. View in web browser. And so here it tells you for each of these And I just ran it on 10 just so we'd have, you know, something to look at. This is the percent duplication rate. So basically for each of those figures that we had before, we have a sort of summary figure. TC content across each of those FASTQ files, millions of reads. Here is that sort of duplicate versus unique read. And actually this is interactive, so you can change it so it's percentages instead of number of reads if you want. I can't remember if there's other, you can configure things here too. Here's the quality by position, and it's a little bit hard to tell, but actually this is laying all the different reads on top of each other. You can see that they're very similar, right? So it's a little bit hard to tell, but you can scroll over and see. I think there's a way to actually change the y-axis. You can zoom in. Oh, you can actually even go up here and like select. Let's see. That work? There. So you can like take out some samples if you want. It's a nice tool. Here's the, you know, the quality per read quality score. Here's that plot of the per base content across the read, and so it's showing it as a heat map. So you can see here it's like kind of gray, which means that it's even, and here you can faintly see that, you know, like these positions have a little bit more green in them, which is G, I think, right? No, A. You know, here it's maybe a little bit more reddish, so it's a little bit more T. Basically, you know, like I said, this is all the plots. The one thing that you get the overrepresented sequences there. And then this is sort of like a summary where it's like those on the side thingy where green is good, yellow is like maybe concerning for each of the different like assays. Questions about that before I quickly move on? Okay. So that's the QC step. So I'm going to go back to, next we're going to do trimming. So I'm clicking on the R cube here to get back to our home base. Go to Assays, RNA-seq Bioinformatics, Notebook, and in case you don't know, you can see here where I am, and we're going to do FASTQ trimming. Anyone need me to do that again? No one's yelling. So, oh, no, someone's yelling in chat. Okay, so if you click on the cube here, that'll get you back to the project, top of the project. Go to Assays, and you can see it's showing assays here. RNA-seq Bioinformatics. Notebooks, and then we're looking for FASTQ trimming. Okay, anyone not have the FASTQ trimming open here? Okay, cool. So I'm going to run this first chunk just to load a few different libraries. And again, this one we're going to, we're mostly going to be using Bash. I'm doing things slightly differently here, which is that here, if you remember, I had a bunch of like, in the FastQC file, I'm setting directories and file names and the number of CPUs and stuff all in this first chunk. For this analysis, the way I have it set up, I have 4 separate files. And if I tried to, so one option would be I could have like basically this sort of information at the top of every file. But if I change something, like for example, the sort of output directory, like if I moved to a new server that I wanted to output to a different directory, then I have to change that in each of those files. And that's, number one, a pain in the butt, and number two, like not so good for reproducibility. And so instead, what I'd like to do is have this separate file, this config, I'm just calling config.r, you can call it whatever you want. This is just an R script where I'm setting up all that information. And then within each of the scripts within this project, we're basically loading that information. So we're using an R function called source that can read an R script. And basically when we run that, you can watch in the environment. Actually, I'll just clean that out. So when we run this, everything that gets done, all the variables that get defined in this config.r are going to be defined in the environment of this this R markdown. So I'm going to run that. Oh wait, actually, did clearing the environment shouldn't affect that, but I'll just do that anyway. Okay, so here I'm sourcing that config file, and so you can see now the environment is populated with all these paths. And so I'm actually not going to use all of this. So for example, some of this information I'm not going to use in this file. I'm going to use in the next R markdown, but I like just having everything sort of defined here, and then I know where to look if I need to change one of those variables. Questions about that? Okay, so I'm just going to create an empty directory for the output of the trimming, and we can do file info and just see that that was made as a directory. So for trimming, we're going to use a program called Trim-O-Matic. There are a few different programs for doing this. We could discuss about what's best. I like Trim-O-Matic. I also use like another version. So there are two different versions of Trim-O-Matic. This Trim-O-Matic SE is for single in data, which is what this data set is. There's a separate, basically the same, but it's a different command. Trim-O-Matic, I think it's PE for, I assume it's PE for paired in data. I can't remember. So if you just run Trim-O-Matic without any arguments, it'll give you a very short summary of how to run it. I have a link here to the manual, which is like kind of confusing, but I've been trying to figure out without the manual. We need to, if we want to do adapter trimming, we have to feed it an adapter file. So that's already made. I have some instructions here about how to make an adapter file and actually a separate script. This is what the adapter file looks like. It's got, it's a FASTA file. It's got the two different adapter sequences, and then it's got the reverse complement, because I never can remember. So depending on where you got the adapter sequence and how the sequencing was run, sometimes you need to give the trimming software the adapter sequence itself, and sometimes you need the adapter sequence as it was synthesized, and sometimes you need to give it the reverse complement. So I usually just like put it all in there. There's not really much harm in doing that. I guess there's a slight risk that you'll like accidentally match sequence that isn't really adapter, but as long as you have your sort of the match length set at a reasonably high number, then that won't be too much of a problem. And so we can do a quick check. We're going to use this program called grep that is going to, here, oh wait, why is it not working? Oh, yeah, sorry, I need to do this at the terminal. Sorry. Oops, didn't like that. All right, oh yeah, okay, I see. That's why that was there. Thanks. Okay, so everywhere here in red, so we're just basically searching through the FASTQ file, and everywhere in red is a match to the adapter sequence. So you can see that we've done something right? We're actually searching for all of these possible options in the FASTQ file. And actually here, we're just searching and counting how many times it matches. And so there are basically 87 matches, and that's perfect matches. Actually, that trimming software doesn't require a perfect match. But there are 87 perfect matches out of, 6,700 reads in this file. Okay, so we're going to run Trim-O-Matic. Because I'm running really late, I'm not going to go through a lot of details here. Actually, I'm going to just start it running and then we'll talk about it. So we're calling Trim-O-Matic. We're telling it that it can use all 16 of the CPUs. So this Fred33 bit here, I didn't tell you this before, but there are actually multiple ways to represent quality scores in a FASTQ file. And they all kind of boil down to whether when you are taking that like capital B and converting it into a number, whether there's one step where you need to either subtract 33 from it or subtract 64 from it. And unfortunately, for a while, Illumina was doing the 33 is sort of the standard, which everyone does, but for a while, Illumina decided they wanted to be different. And so they had the like 64 version. There's no perfect way from data to tell, there's no indication in the FASTQ file whether it's 33 version or the 64 version. And so unfortunately, there are Illumina, I shouldn't just blame Illumina because there are actually other companies that do this too, but Illumina is the most famous one. So there are Illumina FASTQ files floating around that have the sort of the 64 encoding. And so software that's smart actually asks you which encoding to use. And so Trim-O-Matic's doing that. Let's see, you have to tell it, give it the input, the output, and then this really complicated thing is telling it how to do the trimming. So it's saying, here's the adapter sequence. I'm trying to remember, this is something, I have to look this up. Oh, I have my notes here, never mind. So it tells you, let's see, how many mismatches to allow for the adapter, how this we're skipping, and then basically how many bases it has to match. So it has to, it's allowing 2 mismatches, it has to match at least 10 bases. And then this is looking at trimming at the, in this case, Trim-O-Matic is actually doing at the 5' end and the 3' end, and it's, here we're using a pretty low threshold of a Fred score of just a 3 or less. And then this sliding window says like, basically it'll trim if the average quality within like a window of size, let's see, a 4 base window, if the average Fred score is below 15, then you're going to trim at that point. And then this is that filtering steps. So it's saying we're going to throw out any reads that after trimming, they're less than 36 base pairs. So, and it gives you a little bit of, mostly the output you care about is the trimmed FASTQ file, but here it's telling you that like 98.4% of the reads survived, either, they might have been trimmed, but they're not filtered. And then 1.6% of the reads are filtered. And we can just look and see like, here's the trimmed output. And then if we run this again, and search through the output file, now we see that there are no matches to the adapter. So trimming worked. I know that was fast. Any questions? Okay. So actually we have 2 more steps and then we're done. One step's really easy. We're going to go to this genome prep file, .rmd. I'm going to just run the whole thing and then I'll explain to you what it's doing while it's running. You run all. So this is, we're taking the genome sequence and the annotation. So the annotation tells where the genes are and the exons are and the introns are. And we're just processing it through STAR into a format that it can, it's basically can rapidly access that when it's doing the mapping step. So the technical name for this is indexing the genome, but it's basically kind of like doing a mathematical transformation on the genome so that it can do that searching step in an efficient manner. This is a step that usually, like if you work in a lab that say always works with human data, you do this like, you know, the first time you run RNA-seq, or like when there's a new version of the human genome that comes out and you decide you want to use the new version. But you don't do it for every sort of data set. Usually you would do the indexing, you'd keep it around. It's really, because the genome is only 40 megabases, it's really fast for Cryptococcus neoformans. I think this would take like about an hour with a human genome. So it's not terrible, but it's, you know, obviously slower. Yeah, so, and actually in there, we actually downloaded the genome from sequence database. This is just a look at the annotation file. I'm going to skip that in the interest of time. This is the step, this STAR, and then here we're saying run mode, that we're doing the genome generate mode, the genome indexing. I can skip over a lot of those details. And so now we have an indexed genome. That was super fast. Any questions? Ready to go on to actually mapping? Okay, so next we're going to do the mapping.rmd. And again, I think I'm going to run the whole thing and then we can talk through it. So this is that mapping step, right? Where we take each read and we find where it goes in the genome. And then STAR is going to do the counting step too. I'm doing up here at the top, I'm sourcing that config file. And then basically we're just running STAR, where STAR has about the manual of like over 100 pages. There's probably a good 40 or more different command line options. It gives you a lot of control over how you run it. Mostly what we're telling it is we're saying, you know, here's where the index genome is, here's where the FASTQ file is. We're telling it that the FASTQ files are gzipped. And so this is how it needs to uncompress them. We're telling it we're giving a template for naming the output files, because actually for each input file, it generates probably like 6 output files, something like that. We're telling it that we want that count table, and we're gonna generate a BAM file. And we're telling it if there are reads that end up not being mapped, we want to actually hold on to those. Normally, we don't necessarily need to do this, save the unmapped reads, but it helps us for looking at some sort of summary statistics. And this is a brief summary. There were, what is that? 1.7 million input reads. The average read length is 74. Now remember, before the average read length was 76, but we did trimming, right? So we brought down, with trimming, we brought down the average read length. It's mapping, 1.68 million reads, right? So 97% of the reads are being uniquely mapped. There's actually a lot more information. So this is just like, oh no, this is a log file, sorry. There are a lot more details. Here's a little bit of the output, right? So here are the actual counts for the different genes. This we don't care too much about. And here's some more details that it's outputting for us. The number of, it's actually, if I remember correctly, discovering new intron exon junctions that aren't annotated. You have to take those with a grain of salt. Here's some information on the multi-mapping and the unmapped reads. Right, there are some reads here that are unmapped because they're too short. So like maybe we actually should have thrown out I think we threw out reads that were 38 base pairs or shorter, but maybe we should have had a higher threshold.
Was honorable because they tried 290 stop?
No, those are multi-mappers. I think STAR has I think you can control it, but I think it has basically a size limit below which it will not try and map a read. And you can, I don't remember what the default is. We didn't set it. It's just using the default value. And part of that is efficiency, because short reads, it can spend a lot of time trying to find where it maps in the genome. It's a pretty small number of reads, right? Out of 1.7 million, we have 4,000 that are too short. So I don't remember what the default is for STAR. That's something we'd have to dig through the manual for. But so we threw out anything that was 38 or less. And so it's still saying some are too short, so that it must be that it's higher than 38, but I don't remember. So I like to do a little sanity check because I've had STAR, I've never had a problem with, but other software I've had weird things where it loses reads. And so here I'm just checking really quick, how many reads are there actually in the FASTQ file. I'm using a function that comes from an R package called BioStrings. And so there are 1.723745 million, 1,723,745 reads in the FASTQ file. This is the trimmed FASTQ file. And I just want to double check that the number of input reads matches. Like I said, I've had issues with other software, so I like to do that sort of check. Here is looking at, we're pulling out the reads that weren't mapped. And so I will often take these reads and just blast them to see if there's some sort of trend going on, if there's some sort of contamination. Usually these are going to be either garbage reads. You can have just if there was a tiny bit of bacterial contamination at some point in the processing, you might get a little bit of data. You can get skin fragments from whoever, made the libraries and stuff. But, like I said, I'll usually just take these and blast them. I'm not going to do it right now. And then the other thing is that you can actually run multi-QC on the STAR output, which is going to be here. And this is kind of going to be boring because it's just one sample, but you can get the percentage of reads aligned. It's mostly the same statistics where you saw, but it gives you some useful information. Okay, that was a super quick whirlwind tour of bioinformatic analysis of RNA-seq. Any questions? And the most important thing is that we generate one count table. Like I said, I already generated count tables for the full data set, and you're going to use those next week. But you are welcome to, if you want to play with the scripts that are here, you can generate your own data set or your own count tables for the full data set.
Can I ask you a question? In this workshop, what happens to all of these files?
Good question. The Git repos will stay. The Git repos on GitLab will stay around as long as someone doesn't go and intentionally delete them, which I don't expect anyone to do, or if Duke decides they want to stop supporting the Duke GitLab, which who knows whether that'll happen, but it hasn't happened yet. So what happens on DCC is a little bit more complicated. If you are a Duke affiliate, you can have, any Duke affiliate can have an account on DCC. How you go about, so if you didn't have a DCC account before, and you got an account for the workshop, then at some point, probably over the summer, they will get rid of the HIV 2023 group. If your account isn't affiliated with another group also, because you can be affiliated with multiple groups. So like, for example, my account is affiliated with the HIV 2023. I'm in Department of Biostatistics and Bioinformatics, so my account's associated with the Biostats group, and I'm also in the Center for Human Systems Immunology, so my account is associated with the CHSI group, and like there are other things I teach that's associated with. So like for me, when the course group goes away, it doesn't, like my account will still be around, I just, the directory, the group directory won't be around. If your account isn't affiliated with another group, then I think if they get rid of the group, then your account might go away. I think I have, so the safest thing to do is if you want your account to stick around and you're a Duke affiliate. If you're not a Duke affiliate, then it's trickier. If you're a Duke affiliate, and you want your account to stay around, then the best thing to do is to email the research computing group, which I can share the email address, and just say, hey, I have an account on DCC from this, workshop. I'd like to keep my account. Yeah, so the scripts will stick around. And the scripts will stick around forever. The containerized computing environment we use is, I don't know if we have instructions on how to download that, but that's actually publicly available and you can run it on it's super easy to run it on like other Linux computers. It's not quite as easy to run it on like on a PC or a Mac, but it is doable. Yeah, so I talked about this, if you were here for the reproducibility session, I talked about this a little bit. So everything we're doing it in what's called a container, which you can kind of think of it as like a virtual computer. So it's got all the software installed in it. So the software is actually not installed on Duke Compute Cluster. It's installed in this container. And when you go to DCC on demand, and where is it? Like here, for the CHSI RStudio workshop, it's always launching this container that has all the software installed in it. So you can download that container image and put it on another computer, and you can then run, so the container system that we use is called Singularity. So you can run Singularity with that image, and it will start up a container with all the software available. So it's a super easy way to, if you want to like run, basically should be able to run any of the notebooks, any of the R Markdown files on any other computer, with the one caveat that you might have to adjust paths.
So you just download R and Bash.
So you don't need to download any of that if you have the Singularity image. And like I said, I can't remember if we have instructions on how to download the image, but I can definitely post those before the end of the workshop stage.
Yeah, so I actually had a question about that, because I've kind of taken like our lab from like no RNA-seq to like a whole pipeline by setting up like mini conda and environment and all that kind of stuff, and then taking it through that way. So that's why I was asking about the RMD, because I was like, holy crap. And then I was like thinking about how you set up your different environments and things. So it sounds like you kind of run a single container that has all of like your versions and Python versions and all that kind of stuff, and then run everything out of there.
Yeah, and I not to, I don't want to shame you, but
Yeah.
I have a hate relationship with Conda.
I hate it.
Okay, good.
It was just like, I came from a place that was doing, like kind of individual, like module load, kind of this kind of stuff like that.
Yep.
And then I was like,
So
how do I do that
that's
here?
also an option on, so are you running analysis on DCC, or are you running it someplace else?
Yeah, on DCC.
So they actually do have modules on DCC.
Yeah.
I really like using containers because, yeah, oops, I forgot to remind people to the end of the workshop survey. Oh well, too late now. So I like containers because I trust them a little bit more than modules. I trust them a lot more than Conda. I actually have, I'm a little bit embarrassed to say it, although not really. I have built containers using Conda, and especially, there's actually, so you know Bioconda, you probably encountered Bioconda. So Bioconda has a collection for every package that's in Bioconda. They actually have like a container that just has that package and all its dependencies in it.
Oh, nice.
So it's actually nice for, so one way that I like doing, one way that I've started doing pipelines is using either Nextflow or SnakeMake, and I've encountered those.
Yeah.
And so for both of them, you can, for every step, you can specify a container, and they're actually nice. It ends up being a little bit funky, it's not perfect, but the way they're supposed to work is that it will, if there's not a local copy of that container, it'll actually download the container. I can't remember, I think it's Nextflow.
So then you're always getting like the up-to-date.
Yeah, well, it depends actually. You can specify a specific version of a container. So like if you don't,
so if it's not this one.
So if you just don't happen to, yeah. And so like, that's really nice for this, the Bioconda containers, because even though like Conda is like a nightmare, like part of the issue with Conda is just that it's a nightmare because of like conflicts.
Right.
And so like, if you're like just running one step in a container, you're usually like pretty okay.
Yeah, because that's what I was running through, because I was trying to figure out how to do it, transferring from the module load kind of like job to the mini conda things, and then I had to figure out which the issues were, and then go through and create different environments, solve those issues, and so on.
So
it was a big pain.
I can point you to, if you're interested in building containers or using existing containers, I can point you to some information. I've thought about teaching a workshop on containers, but I've never gotten up the nerve.
I'm a little familiar with containers, like I run a containerized system at home, kind of to control like different parts of the home and things.
Mhm.
So like very briefly, like dipped my little toe
Yeah.
into it, you know, but I like conceptually it you know, it all makes sense. So,
Yeah, but if you want information on like Nextflow,
Yeah.
like how to do those pipelines, I can point you to some examples.
okay.
And like I said, I can definitely point you to the container that we use for this workshop. It's probably has everything that you want for like basic RNA-seq.
Do you know the container name off the top?
So it's,
Or if not, if I can
so
find
it's
it.
easy to find, or kind of easy to find if you go to, you know how to get to the Git repo?
Yeah.
So it's in a group that's called, I think, R20 something, R25 HIV something or other.
Okay.
And then there are a bunch of repos in that. Basically, we have one repo for every year that we've done this. And then there is, I think we're using there's a container image.
Sorry, I don't want to take up your time. If you have
No,
to go,
that's
I can
okay.
look
I'm
around.
just, I'll just have an excuse. Wait, why is this not working? I swear it was. So actually, so the image is actually already on DCC.
Oh, okay.
So that's
So
one
theoretically, I
place
could
to find
just
it.
kind of
but,
like pull it.
yeah, but I'll show you. Well, I'll show you the two places to find it. Okay, so here's how I always find it: is I find this one, and so if you go to there, then you can click on this, which is the group, and it's this singularity image. So we're basically
okay.
it's mostly the same one from last year, but we just versioned it.
Okay.
So this is the singularity recipe, but further down here it tells you how to pull it. So you can do singularity pull, and the version. So actually the one that we use today, it has some changes that we haven't. So if you go here under tags, so this is yeah. So if you do, I don't think this is today's. So you can pull it. So like this is if you actually do, where is it? Yeah. So if you do this right now, it's latest, but we'll sometime probably in the next few weeks we'll actually tag it with a version number, and then you put the version number there.
Okay.
And then the other thing is that it's that's the most reliable thing.
Okay.
Yeah, we've been Tyler, who's teaching in 2 weeks, 3 weeks, is I think probably still adding some packages. So then you might not remember this, but I can email you if that's helpful. Studio. Okay, so then it is right now in this directory. It's this one, but we'll probably update it.
Okay.
So you can see that we have version 44 in there and version 43 in there.
Okay.
But every once in a while they kind of clean up here. But that's on.
So that's an image that's curated kind of by DHVI or
this image is it's curated by like me and Tyler and maybe a few other people.
Okay.
And so these are like the way they've set up these directories. So there's a nice system for building singularity. So you can build singularity images on DCC, but there's also a nice system within Git. Within the Duke GitLab that they have this continuous integration, continuous development set up where did it go? Here. So this repo just has 3 files, right? That's the singularity recipe. So this YAML defines, and this is kind of like mostly like a off the shelf that like one of the research computing people developed. I modified it a little bit, but basically what it does is when you push to the repo, if there are changes to the singularity recipe, it runs a build job, and then it like puts the built container on some server somewhere that is where this pulls from. And it actually has like there's like a separate version where if you tag it, if you tag a commit, then it will build it and then name it with the tag, right?
Got it. Okay.
So I actually usually when I build images I use and so you don't. I'd have to find the original version of this CI/CD YAML, but it's I like how it's set up because it like just kind of builds on
built on the back end and then
you can pull it down from that like repository. So that's like where that's sort of the public repository.
Right.
I'm hoping so far they haven't like said like, oh yeah, you need to clean you have a bunch of images there you need to clean up, but it lets you basically you can
then distribute it.
so far like have like any tagged version is up there for the moment.
Okay. Cool. Yeah, that's really
it's a whole can of worms.
Yeah. Because I was like going through it and kind of as I'm trying to bring this to people that are like some people in my lab are not going to be the most able to kind of go line by line and make their changes. So I'm kind of thinking about how to make this as spreadable to other people.
Yeah, and it is definitely trying to get pipelines so that they're usable by people who are not super savvy is very challenging.
Yeah. So awesome. Well, thank you so much.
No problem.
I really appreciate it. See you next week.
I'll be back in 2 weeks.
In 2 weeks.
If you want to talk more.